All right. So, how are you today? Today is yet another look at molecular genetics, SIL1001. Now, in the previous lecture, we stopped at looking very quickly at translation. Maybe I can just do a recap on that one. If you have any questions, please let me know. If I don't see your text, you can just you can send your questions through chat or even WhatsApp. I've got both screens here. If I don't see them, you can just turn on your mic and call me. Right. So okay. There's still students coming in. So anyway, things that is important for you to remember. Well, the take-home message for translation is, this is the first slide that shows you the different ribosomes. On the left is the ribosomal unit for prokaryotes, and on the right is the ribosomal units for eukaryotes. A ribosome molecule, if you know the small subunit and the large subunit. I'm still letting people in. The small subunit and the large subunit, right, is actually those molecules are actually made up of a combination of proteins and an RNA molecule. In this case, is ribosomal RNA. So you should be introduced to, there are the major types, 3 types of RNA: the mRNA, which is the message, rRNA, ribosomal RNA, and then the transfer RNA. So the rRNA is here. And then, when you see one of the first steps in translation is this. I'll get back to the slides. One of the first steps of translation is this: the binding of the small subunit to the mRNA. And this binding, remember, the small subunit is also made up of RNA and proteins. The actual binding between these two is actually interaction between the rRNA and the mRNA. And the interaction occurs within the sequence that is called the Shine-Dalgano sequence. This is what happens. So you have the mRNA, and then this is the ribosomal RNA. And this is called the Shine-Dalgano sequence. This is the site of attachment, all right? Other characteristics of translation. Okay, the tRNA, special characteristics of the tRNA, it has special bases. By that I mean you normally have the A, G, C's, and U's, but for the tRNA, they have special bases. Look at here again. You have the I, which is the inosine. You have the Psi. You have the DHU, which is dihydrouridine ribothymic. So these are special bases and special bases which are characteristics of tRNA molecule. What is important? It's important for its function, and the most important function of a tRNA is to become the molecule that is connecting the mRNA language, the codons, with the tRNA will bring the anticodon, and this on the other end will bring the amino acid. All right. Okay. This slide talks about tRNA activation, or sometimes it's called amino acid charging, tRNA charging. There are several terms to it, but basically what it's doing is putting the you know there are 20 different amino acids. You have many tRNAs, right? tRNA bringing anticodon, and then so you have to match the right tRNA with the right amino acid, and this is done by amino acyl tRNA synthetase. And this step is very crucial because once that is done, I mentioned this in the previous lecture as well. Once you have tRNA that is fixed with the amino acid, in the process of translation, the there is no system to ensure that the tRNA brings the right amino acid. What only happens in translation is that you have the mRNA, and then you have the IFs, or the initiation factors or the elongation factors that's going to bring the tRNA to match the mRNA. That's all, right? It doesn't care whether it brings, because there was an experiment conducted where they actually changed. So this slide says there was a change. They changed if you have all right, if you have a tRNA and then the tRNA is attached with the correct amino acids, they change the amino acid. In this case, it's by from cysteine to alanine. They find that the product of translation, all the cysteines have been replaced with alanine because the translation system doesn't check for amino acid. So it's very important that the function of this amino acid tRNA synthetase, which matches the correct tRNA to the correct amino acid, this has to be tip-top. There cannot be problems here. All right. Other important take-home message. Well, this is the process of translation. You can read through. They are self-explanatory. And how does it stop? By what they call as release factors. These are the release factors. When it reaches the stop codons, then there are no tRNA for stop codons. What they have are what is called the RFs, release factors that recognize stop codons, and then they bind to the stop codons, and then this gives a signal for ribosome to stop translation. And then this is where also the new polypeptide is being released. Okay, so that should take care of translation. And then a little bit on the genetic code. Now, some characteristics of the genetic code itself is made up of triplets, 3 bases to 1 amino acid. You have A, T, Gs, and Cs, right? You have 4 bases. In each case, you use 3 base. This is what we call as the codon ratio. So 3 base for 1 code. So it can start with AAA, AAT, AAG, AAC, and AT, AAT, and so on. All right. Okay. So that's it composed of triplets. Non-overlapping. In general, it's non-overlapping, but you see in some viruses there are some overlapping codes, but we're not going to look at that. So in general, you know that the codes are non-overlapping. It just says that when you start reading the translation, you just read 3, 3 by 3, 3 by 3, 3 by 3 by 3 by 3 by 3. You don't read 3 and then you add 2, and no, it doesn't do that. So this is a very fixed reading frame. It's comma-free. There's no degenerate. Somebody asked me about degenerate. Degenerate just means that some amino acids can be coded by more than 1 codon. For example, leucine, serine, arginine, each of the amino acids has 6 codons. Methionine has only 1. Isoleucine has 3. Histidine and so on, they have 2 codons. Yeah? So that's what it means by degenerate, 1 acid amino with more than 1 code. Collinearity between, it means that the DNA will determine the mRNA, and then the mRNA will determine the amino acid. If there's a change in DNA, then mRNA will change, then also the amino acid will also change. So this is the collinearity. There's always a consistency between DNA, mRNA, and then the resulting amino acid. That's where we study mutations. If there's something wrong with the amino acids, you can actually trace it in the DNA. All right? Okay. It's almost universal. That means codes for organisms as small as the virus and the bacteria, the codes used in those organisms are the same coding system that is used in the elephants and the whales, the biggest mammals and the biggest animals, and birds, and all organisms use the same code, but the key word here is almost. There are small differences, especially when you look at mitochondria. In humans, for example, the mitochondria has 4 different stop codons, and UGA is not a stop codon in the mitochondria, right? But these are, again, very, very minor differences, but in general, the genetic code is universal, almost universal. This is what it means. Moving on, I'm going to skip this slide because this, if you follow on further with the course called Molecular Genetics, you are going to look at the deciphering of the genetic code. These are just explanations of the genetic code, where you have replication that copies the DNA, and then you have the expression of the genetic molecule, where you have the DNA, mRNA, and then the protein, right? This is what I just mentioned. What is the coding ratio? Do not get confused. These are just examples. These are just hypothetical situations. This is because they're trying to describe what these people were trying to do. You know, Karana, Ochoa, and Manago. These are people who was very instrumental towards deciphering the genetic code. This follows on from, you know, the historical aspect, when they find, you know, remember Avery McLeod McCarthy provided an experiment to show that this is the DNA is important. The DNA is the genetic material. And then other experiments like from Hershey and Chase and so on. So those are the experiments. Then they went on to investigate, right? Shargoff says it's double-stranded. Watson and Crick came up with the double helical structure. You know, so the progression until they realize it must be the language of the DNA. Then these are the people that is instrumental in deciphering the genetic code. Okay. Matthew and Nirenberg, not all of them, some of them actually received Nobel Prizes for their effort. This is the general, in textbooks, in most places, the way they present the genetic code is almost the same. It's always UCAG, UCAG, and then the third one is UCAG. It's always like that. And then from here you can see several characteristics. I'll go to the okay, before I go into the code, again, these are some characteristics or properties of the code. There are 64 possible codes because you have A's, G's, T's, and C's or U's. You have triplets. That means you have 3 positions. How you can calculate that? You have triplet code, right? So you have the first base, second base, third base. For first base, you have 4 options. Second base, you have 4 options. Third base, you have 4 options. So 4 times 4 times 4, you get 64. But in the 64 codons, 61 encodes for an amino acid. 3 is the stop codon. UAA, UAG, UGA. The ochre, amber, and opal are specific names for these codons. Right? They are called nonsense codon, they're called termination codon. So these are the different names, or sometimes stop codon, and AUG is the start initiation codon. Again, you'll find that in this introductory level, if you read textbooks and so on, you'll see that these are some of the rules. But if you go further into research, you know, you go to high level of molecular genetics, then you'll find that AUG is not a start codon. Sometimes it's GUG, sometimes it's ATT, you know. But in general, it's AUG. So at this point, at this stage, is whatever is given to you should be sufficient. All right. Degenerate codes. Every amino acid are coded by at least 1 codon. At least they must have a codon, and methionine and tryptophan have 1 codon each. Now let's just look. Okay, here I've divided this. You'll see the table can also be there's a characteristics to the table. You'll see here because of the arrangement of the UCAG, UCAG. You see phenylalanine. Now look at this, right? Look at this. The pink one are the codes where the amino acids are determined by 4 different codes, like proline, CCU. So proline is actually determined by CCN. N means any code, so it can be CCU, CCC, CCA, CCG. Or threonine is ACU, ACC, ACN. Alanine is GCN. Valine is GUN, and glycine is GGN. All right, so that. Now look at this column, the A column. You'll find that the amino acid is determined by codons, and it depends. The difference is whether the codon ends with a purine or pyrimidine. You know your purine and pyrimidine? Cu is pyrimidine and Ag is purine. So if it is Ca pyrimidine, is histidine. Ca purine is glutamine. Aa pyrimidine is asparagine. Aa purine is lysine. Right. So for this code, all four for this column, all 4 determines the same code. But for this one, it depends on whether the last base is a purine or pyrimidine. You also have the oval blue codons that have 6. So you have leucine that's coded by CUN and also UU. So this is all leucine. Serine is 4 here and there's 2 here. So UCN is serine. AGN is also serine, and arginine is CGN plus AG. So there are some characteristics. Okay, so this is a characteristic, and here the generic codon. What it means here is that some of the codons have 2, some of them have 3, 4, and some have 6 codons for each amino acids. All right. Moving on. So these are again the characteristics. What I mentioned earlier is non-overlapping. There's a specific reading frame. It starts off and then it just goes on 3 by 3 by 3 by 3. So you don't skip, you don't miss one or anything. And this is the basis of mutations as well. This is where this slide talks about the fact that in some instances the genetic code is not universal, right? Codon UGA in yeast mitochondria, in human mitochondria, UGA normally in the normal cells is a stop codon, but in the mitochondria is tryptophan. So very, 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 very small, very, very minor differences. Moving on to mutagenesis or molecular basis of mutation. Mutation is a heritable change, right? Gives you a mutant. If you like to watch as X-Men, those are mutants, but those are good mutants. But in our case, when we study mutants, it's always mutations cause diseases. Mutation is always seen as something that's negative, right? So mutation is a heritable change. There are certain terms to mutation, point mutations, where the mutation involves a very 1, 2, or even up to 5 bases. Very small region is called a point mutation. If if the mutation is larger, then it's called a gross mutation and so on. So there are different terms. That the terms that you're going to see in this is point mutations. Now, here you talk about transitions and transversions. Transitions are changes between the same class of bases. Purine to purine, pyrimidine to pyrimidine, right? So a C to U and A to G, these are all transitions. So C to T or U, C to T is transition, A to G is transition, G to A is transition, T to C is transition, right? But a change between one class to another is called transversion. So from A to C, this is a purine to pyrimidine, it's called a transversion. C to A is also transversion. So in this figure, the blue arrows are transitions, the red arrows are transversion. It also means that there are more transversions compared to transitions, correct? Right? But in actual case, I mean, you would expect to see a lot more mutations on transversion than transition, but actually there are more mutations on transition because a lot of mutations actually cause transitions. We'll talk about that when we talk about mutations. Sorry, I need to switch on my power source. So mutagenesis point mutations involving either 1, 2, 3, or up to 5. And this mutation can be either a change or replacement. That means A change to G, G change to C. You know, those are transversion or transition. These are called substitution. Or you can lose bases, which is deletion, or you can find additional DNA, which is an insertion. So three main classes of mutations are substitution, deletion, and insertion. Now, it's just a point to note, later when we talk about DNA repair, we see one of the mutations, and like, let's say there are 4 bases, right? Exposure to UV causes thymine dimers. I don't know whether you've heard that. Thymine dimers, that means if you have T and T side by side, if you expose them to UV, they'll tend to be close together. They tend to be attached. This is called a dimer. And this is a mutation. Why? Because if you imagine, if these are 4 different bases, and then you have the polymerase trying to make, in the process of replication, trying to make new DNA, right? So it will look at the first base and then add the complementary base and so on. But if this is attached together, right, there's a chance that when polymerase reads it, then it doesn't know whether there are 3 bases or 4 bases here, because now the shape is distorted. So that's why if it misses and thinks that there's only 1 T here, so then you get a deletion in your replicated product, right? Sometimes the skew, the polymerase thinks that the distance is not usual between these 2 bases. It's too big. Maybe there's a missing base. So sometimes the polymerase will add an additional base. So this is one of the causes of addition and deletion because the shape of the DNA itself has been distorted. Moving on with the mutagenesis, what can cause mutagenesis? A few things. It can be divided into physical harm, chemical harm, and enzymatic harm. Physical harm is, you know how the chromosomes undergo pairing in mitosis and meiosis. They go pairing, they cross over, and then they will be pulled apart. And sometimes it doesn't go in the normal fashion. Sometimes, it gets tangled, and because of that, it may pull too hard and you get chromosomal breakage. And so this is what you call as physical harm to the chromosome. Chemical, of course, if you are exposed to certain types of chemical, I mean, there is the whole mutagen class of mutagen. We are not going to do that in this lecture, but basically there are chemicals that sometimes a chemical that can add methyl groups, that can remove methyl groups, some DNA that some chemicals that intercalate. What does intercalate? That means, you remember the structure of DNA is double helical structure. It's got the major groove and minor groove. Some chemicals just like to insert itself within the grooves, and because of that, it causes tension to the DNA and changes its structure. And remember, I said in the previous, just a few minutes ago, if there is a change in the DNA structure, it will result in the polymerase not reading it well and probably treating it as a problem, and the result is probably a mutation. Okay, there's a question. Is the transcript mRNA anti-parallel to the DNA template? Yes, because you see, that is the general rule of DNA binding, right? Anytime you see a binding between 2 different molecules, it's always anti-parallel. So if this is your DNA, right? If that larger green is 5' and that is 3', all right? You will, so your molecule, if there's 5' to 3', remember synthesis must always be 5' to 3'. So if there's 5' to 3', so it will synthesize 5' to 3' that way. So it's anti-parallel. It's always, even the codon, anticodon, right? AUG in the codon, then you'll have CAU, all right? AUG, you don't, the anticodon is not AUG, it's not UAC, it's CAU, okay? So because you're reading it, this is 5' to 3', the anticodon is also 5' to 3', right? Okay, I hope that that's clear. And for those of you who have joined in, please make sure you put in your name and matrix number for attendance purposes. Right. Okay, so these are the type of mutations. Okay. So you have, because DNA or mRNA brings message, it brings a code. So if there is a substitution, if there's a change, if there's a deletion, loss, if there's an addition, you insert something, it changes the meaning. All right. So these are some different what should I say? Different outcomes of mutations. You have, sometimes you have same meaning. Why? Well, because remember, some codons are degenerate. CCA has the same meaning with CCC, has the same meaning with CCG. So that means to say, if you have a codon, a CCA, if you change the last A to a G, you still get proline, correct? So that case is the same meaning, or silent mutation. It's a mutation, but it's silent. Silent means it doesn't have that obvious effect. Sometimes it changes, right? The way to know this is to actually refer to the genetic code, right? If you look at this code, if you want me to enlarge this, okay, you will find that, look here. Say, for example, look at this CA, right? If the U changes to a C, well, the code is still the same. CAU, change mutation to CAC, it still codes for histidine. But if CAU changes to CAA, then it changes the meaning. So it depends on so the outcome now, if it is the first change, U to C is a silent mutation, but if it's U to A, then it's a substitution. All right, so so things like that. Okay, so that's how you determine what type. I have other examples I will show you in a bit. Right, so these are the categories of mutations, either silent or the substitution, the missense, that means the meaning is already wrong, or sometimes there's no meaning, nonsense. Sorry, I have to go back to the genetic code again. Let's say, for example, here, all right, UGC, which codes for cysteine, up here, top right corner, UGC codes for cysteine, and the last C changed to an A. So now, instead of it reading UGC, it reads now UGA. UGA is a stop codon, and that is classed as a nonsense. Okay, sometimes also called a null mutation. Okay, here is the example. This is the original, right? The original CGA. If there is a transversion, that means the T changes to an A, right? So CGA changed to CGU is a silent mutation because it still codes for arginine. CGA, if the middle bit undergoes transition, CGA becomes CAA, then the code from arginine becomes to glutamine. So this is a missense mutation. CGA, if the first C changes to a U, CGA becomes UGA. This becomes a nonsense, a transition as well. So this one becomes nonsense mutation. So this slide tells you the possible different types of outcomes if there is mutation. It's not saying that all three is going to happen, but depends which one is occurring, and it will tell you what type. Okay. This is a more friendly kind of look at mutations. If you can see, so it's written in such a way that this is just an example, yeah, an analogy. So if you had a bad red dog and the cat, if the B changes to an S, changes the meaning, right? So the bad becomes sad, the sad red dog and the cat, and then again, if the G changes to a C, it gives you a different meaning as well. If there is deletion, that means that D has been removed, one base, removal of one base. This is again within the point mutation. Then because the reading of the DNA molecule is continuous, right? It doesn't jump. So you remove one, then it becomes BA. Remove D, right? Look at this is the original sequence. If you remove the D's, and then becomes THE-BAREDD, OGA-NDT, right? So the whole, and this is called frame shift. That means the reading frame has shifted. It can happen either if you take out and also if you put in. So here is also another one in the example where an N has been put in from the original. So then it makes the whole reading frame jumbled up, right? Okay. This is a more this is a genetic example. So this is the wild type. Wild type is the normal sequence. So methionine, tryptophan, alanine, valine, proline. If the GCA changes, always you always refer to the wild type. Yeah. So if that GCA changes to the GCT, alanine still means alanine. So this is a silent or synonymous mutation. If that TGG changes into a TGA, TGA is a stop codon. That means it stops there. Right, so now you have a shorter protein. Remember, in translation, ribosomes will read codon after codon. Usually, it will read TGG, but there's a mutation to TGG to TGA. So if it reads TGA, then says, "Oh, I need to stop now." So you have, as a result, you have a protein that is shorter than expected. If the TGG changes into the TGC, it changes the meaning. So instead of having methionine, tryptophan, alanine, you get methionine, cysteine, alanine, valine, proline. That's a missense. If there is an addition of one G here, you add one G here, TGG, GCC. You know, this is a frame shift, and then changes the meaning also. Methionine, tryptophan is still the same, and then it changes from alanine to glycine, valine to serine, and so on. If you minus one, yeah, this is actually what I do for a living. I'm trained in human genetic. I did my PhD in mutation detection. So this is what we do. If we have patients, you want to know what is the mutation, we take the DNA out. Well, we extract the DNA, we do some PCR, do some sequencing, and I will start reading and then trying to see what is the mutation. And then once we have the mutation, I will give the information back to the clinic, to the clinical doctor, and then they can do, you know, diagnosis, prenatal diagnosis. That means, you know, so things like that. So this is basically what my hobby, doing research in mutation screening. Okay, so then that is the second part of the course, the lecture finished. Now, I'm going a bit fast again because I need again. Don't worry, because all this are going to be on YouTube. Also, I've also made videos of each of the topic on its own. So it's up to you to watch which one suits you well, and you can still ask me questions. It's 11:45. We have a few more minutes before we finish. Now we are entering gene regulation. Now, for gene regulation, there are a few key points that the purpose of this is, one, to tell you the fact that genes are regulated. Some genes are not. So you have two categories is inside this slide. So some genes are always on. That means these are genes that are required for the cell's day-to-day function. You know, definitely it will need that gene product. So this is, so you don't need to switch on and off. So that is called constitutively expressed genes. So genes, you have the constitutively expressed, and then you have the regulated. Now, regulated genes, there are some genes that are inducible. Basically, it means it can be switched on if you need it. Inducible, you can induce it. Or you can repress it, that you can switch it off if you don't need it. Okay? Constitutive, always on. Regulated can either be inducible or repressible. Inducible means if you need it, you switch it on. Repressible means it's always on. If you don't need it, you have too much of it, you switch it off. So these are the two different categories. Positive and negative control is how this gene is being switched on. We imagine you're in a car or a bicycle and downhill. If you want to move, you know, you pedal or you put on the accelerator. But at the same time, if you don't put the accelerator, if you just remove the brakes, it will also slide down, right? So both effects give you a similar outcome. Accelerator will bring you forward. Taking off the brakes will also bring you forward. So this is positive, this is negative, but the outcome is still the same. We'll see that in the next slide if you're a bit confused. Down here just gives you some terms that you're going to see. Effector molecules, these are molecules that involve in gene regulation. You have the inducers, repressors, and whatnot, activators. Allosteric transitions, where proteins, if you view proteins in a three-dimensional manner, right? You know, when you do translation, you have a string of amino acids. This string of amino acids will then be folded into a three-dimensional manner. Now, because it's folded, the folding is based on interaction between molecules. And sometimes the interactions can change, that they fold differently. Different folding will give the protein different properties. For example, if you're going to see this later, okay, this is an example. Now look at this green molecule. This is a repressor molecule. In activated situation, in this situation, in top situation, it binds to the DNA. All right, so this is the repressor. Actually, it's a dimer. That means there are two molecules of repressor binding to the DNA. But if you see this, there's an inducer molecule here. If the inducer binds to the repressor, it changes the shape of, this is still the same repressor, but now it has changed the shape. And because it has changed the shape, it doesn't really now bind to the DNA anymore. Instead, it just binds to itself and just floats about. This is what we refer to as allosteric transition. That means binding of something to a protein changes its shape and then changes its property. In this case, initially it likes to bind to DNA, but binding with an inducer changes its shape, so it doesn't like to bind to DNA anymore. All right, okay. All right, so that is allosteric. And then operon just talks about the structure of how DNA genes are organized, and then factors and elements are, you know, how gene, if you look at transcription, you have the binding of polymerase and sigma factor to the promoter sequence and an operator. So promoter and operator are what you call as elements. This is an inducible system. On the left panel is a negative, on the right panel is positive. There are just two examples. Here you see, remember inducible, it can be switched on. You can switch it on by removing the repressor, or you can switch it on by adding the activator. Correct? Both cases, left and right, results in switching on. So that's where you get negative control, that's where you get positive control, just different terms to refer. The next slide talks about repressible system, that means switching off. So you have either, the negative control is the putting in the repressor molecule, and then positive is you remove the activator. So basically positive, negative refers to the molecule that has a negative impact, repressing, whereas positive talks about a molecule that has a positive effect. But both can be used to control, to shut down. So a repressible system, if you remove the protein that is giving it a positive effect, you remove it, then it will switch it off. But repressible system, if you add a break, then it will switch it off. So again, right, the outcome of inducible is switching on, the outcome of repressible is switching off. All right. This is an example of an operon. An operon is an organization of genes. Normally we would expect one gene to have one promoter and so on, but they found that in bacteria, because the bacteria is such a small organism, it has, it needs to save space. So instead of having every gene its own promoter, any gene or group of genes that is related in terms of its function, they can group together, right? It's the same thing as the lights in the lecture hall. You have one switch that controls many, many lights. You don't have one switch for one bulb, one next for next bulb, and then you have like a whole wall of switches, right? So no, you don't, because you know that when you switch on the light, you need a few lights to be on, so you just need one switch. So this is the same thing of organization of genes in this case, where if you want a function, you need to have this gene A, gene B, and gene C. SG is structural genes. Structural genes are just genes. Doesn't mean that it has to function in structure, but it just means that these are genes that gives you a protein product. So all this SG1, SG2, SG3 is controlled by one promoter and operator. All right. And then also, it is also regulated by the regulator gene. So this is the operon. And then this group is the whole control system. This is an example of how an operon is induced, right? We've seen it already. So initially, it's normally off. So there's a repressor molecule present that it binds to the promoter. So that means your polymerase cannot bind to the promoter region. It cannot bind it, it cannot start transcription. But if you remove the repressor, how do you remove the repressor? Because you have inducer. Remember, inducer can bind to the repressor and causes allosteric changes. It doesn't. So now the repressor doesn't bind there anymore. And now that the repressor hasn't bound, it's no longer binding to the operator and promoter, polymerase can bind and start transcription. I can ask you a question. Is this situation in the prokaryotes or eukaryotes? Well, the answer would be prokaryote. Why? Because you can see as the polymerase is transcribing, making transcription, that is already okay. You can leave for those. Some people have tests at 12, so you can leave. Yeah, it's all right. Again, all this will be recorded, so it will be posted online. So you have ribosomes. Transcription and translation occurring together means that this is coupling, coupling of transcription and translation. So this is a prokaryotic system, and this is an example of repression where again it's normally on and it switches off. Okay. I think yeah, because it's close to 12, I can call this a day. Hang on, let me see. All right. Yeah, some of you have classes and you can leave. So we'll call that a day. But just to a little bit of an overview. So we will have this. We are going to look at 3 different oprons. You are going to look at like opron, tryptophan opron, and then arabinose opron. All these are already in YouTube. The links are already there. You can have a look at the videos, and then finally, then we should be able to finish all the lectures on recombinant DNA technology by next week. Okay. All right. As usual, if you have any questions, you can post it out. I can answer your questions. If not, thank you for your attendance. All right. I'll see you in the next lecture.